Hello, everyone. I'm Huyen Bui from Montana State University, and I would like to thank the organizers for letting me present our study on bacterial-algal interaction in high pH, high alkalinity algal conservation. This study is carried out at Montana State University with collaborators at University of Toledo, Ohio. And it is funded by the Department of Energy together with support from JGI and PNNL via a ficus project. So why are we studying algae? Algae are attractive biofuel feedstock because they can fix carbon from CO2 and then convert the fixed carbon to hydrocarbon fuel or algal oil. And this oil can be used as a direct replacement for petroleum fuel. Compared to plants, algal potentially have higher biomass yield with similar or higher energy content. Moreover, algae can be cultivated in marginal lands or low quality nutrient and water, so they don't compete with food and feed production. However, it is still too expensive to produce algal biofuel because current cultivation technology requires concentrated CO2, which means that you can only grow algae in a small fraction of suitable land where CO2 sources is available. On top of that, if this CO2 must be captured and parsed into the pond, and the cost of CO2 supply is estimated to be about 20% of the production cost. Another challenge for growing algae is that the algal cultures are very frequently infested with grazer, bacteria, viruses, and non-productive algae. These infestation can lead to culture crashes that can cause up to 30% loss in biomass. Excuse me. So for the last 10 years or so, our team has been developing a new approach to grow algae using high pH, high alkalinity conditions, using a strain of chlorella isolates from Salt Lake, Washington, where pH is around 10. We was able to sustain productive algal growth without concentrated CO2. Moreover, the high pH, high alkalinity media can capture CO2 directly from the atmosphere and prevent culture crashes due to predators. Interestingly, even in this extreme pH and alkalinity, the algal cultures are not free of contamination. Since 2019, we have routinely analyzed the microbial community in the culture by sequencing the V4 region of the 16S rRNA genes. We found that SL804 cultures recruited diverse bacteria communities. And here's an example where the culture contained more than 30 bacterial genera. This observation is actually not that surprising because in natural habitat, including alkali lakes, algae almost always co-occur with other microorganisms. Some of these organisms can be agicidal, so basically they feed on the algal cells, but there are also beneficial bacteria that can provide algae with essential nutrients like vitamin or facilitate the uptake of nutrients like iron. Actually, algal and bacteria interaction has been likened to the relationship between plants and soil bacteria, including the symbiotic interactions between root nodule bacteria with plant. These bacteria provide nitrogen to plants in exchange for carbon. So our goal is to understand these interactions between bacteria and algae so that we can promote a beneficial interaction. With the support from JGI via ficus user proposal and from several Beto grants, we are using multiple omics approaches to study spatial relationships, the metabolic potentials that encode in the genomes, the gene expression profile of the community, and the metabolic interactions between bacteria and algae. And I would like to share some of these exciting results with you. So we use high resolution microscopy to look at the spatial relationship between bacteria and algae. On fluorescence microscopy, we observe that bacteria in this culture can be free floating or associated with algal cells. We then send this sample to PNNL for helium ion microscopy and TEM. These images show extremely incredible detail of this association. It showed that bacteria not only attached or sticking to the algal surface, but they actually make contact with the surface, and potentially have some exchange of material through this interaction. On TEM, we were able to find examples of what look like a bacteria cells that burrowing into the outer membrane of an algal tetrad. So to explore this interaction, we sequenced the metagenome to understand the bacteria taxonomy and the metabolic potentials. So in discussion with folks at JGI, they pointed out a concern when we trying to sequence the algal bacteria genome, which is that the algal genome is larger than bacteria genomes. And algal cells also contain many copies of mitochondria and plastic genomes. So the concern is that SL804 DNA may overwhelm the DNA pool, leading to very low sequencing coverage of bacterial genome. So to test this, we decided to run a pilot metagenome using a maintenance culture with low bacterial diversity. And based on the cell count, we estimated the ratio between algal and bacterial cells is 1:1. When the data is available, we tested this by aligning the reads to SL804 genome that we sequenced independently. And as you can see in this table, between the two replicates, up to 93% of the Illumina reads were aligned to the algal genome, and leaving only around 10% of the reads for the sampling of bacterial genomes. But we went ahead and assembled a metagenome anyway using these bacterial reads. We use MetaHits to assemble the metagenome using and being the context with concogs. After that, NVO was used to curate the beans, estimate the genome completeness, genome coverage, classify the taxonomy, and annotate with tag numbers. So using this pipeline, we identified 8 bacterial mags with completeness of greater than 94%, and we calculated the relative abundance of each mag by dividing the coverage of individual genome by the total coverage of 8 mags. So you can see here that only 8 genomes are present in replicate 1 and 2, but the relative abundances are slightly different. So this shows us that it is important to sequence multiple replicates if possible. Now that we know who they are, we can look into the genomes annotation to determine their metabolic potentials. As I mentioned at the beginning, one way bacteria can benefit algae is to provide vitamins, especially vitamin B, that is essential for algal growth. So I checked the tag annotation to see if any of our genome encode for complete B vitamin synthetic pathways. And the answer is yes. Most of these among these genomes, we found complete pathways for synthesis of B2, B6, B7, B9, and B12. At the same time, there's no complete pathways for any of these B vitamins in the genome of S0804. So this finding strongly suggests that the bacterial community may provide B vitamins for S0804 growth. So at this point, we had observed intimate association between algal and bacterial cells. We had sequenced the metagenome, one pilot metagenome, and showed that some bacterial genome encode for synthetic pathways of B vitamins, which they can provide for algae. We're now in the progress of making metabolic models to further understand their potential benefits. At the same time, we had also tried characterizing the gene expression profile by meta-transcriptome. Unfortunately, the same story as the metagenome, the algal RNA dominate our samples even more than in the DNA samples. And we got very, very low coverage of bacterial transcripts to make any meaningful analysis. So now we have to explore alternative approach. One of them is isotope labeling, quantitative proteomics. For the metabolic interaction, we started using BondCat to characterize the active bacteria. In BondCat, the culture is incubated with a methionine analog called HPG. When enter the cells, HPG substitutes methionine in newly synthesized proteins. And these new proteins can be stained with a dye that binds to HPG. As the result, the active protein, the cells with active translational protein are fluorescent. Then the label cells can be separated using FACS, and we can identify their taxonomy or sequence their genomes. When Isaac Mueller, a PhD student, performed BondCat on our culture, he found that BondCat only labeled active bacteria, as you can see here in yellow, but not the algal cells, which is visualized by chlorophyll autofluorescence. So it is possible that HPG or the dye used in BondCat cannot penetrate the algal cell wall. This is unexpected, but it's very useful because it allows us to stain the bacteria and sort them without being interfered with the signal from algal cells. Now, Isaac is using this method to collect active bacteria populations in different growth conditions and ask if they are different in taxonomy and the level of association with algae. I don't have time to go through this study, but we'll present some of the data in my poster. So please stop by on August 30th. So looking ahead, the studies I presented today allow us to start designing experiments to correlate the spatial relationship with activity, which can be provided by BondCat signal, and the identity of the microbe provided by FISH probe specific for each genome. We also plan to use nanosims to detect the exchange of elements like carbon, nitrogen, iron within the population. In a preliminary study, John Cliff and Sarah Lecty at PNNL can measure the incorporation of carbon and nitrogen in bacteria and algae over time. In conclusion, I would like to leave you with some take-home messages. It is likely impossible to prevent invasion of algal cultures in open ponds, and it might be necessary to embrace the role of microbial communities instead of trying to maintain peer cultures in industrial setting. And we believe that integration of omics approaches will help characterize the potentially beneficial interaction between algae and bacteria, and insights into how to control the community are critical to improve the performance of algal culture in long-term cultivation. With that, I would like to thank our lab members, collaborators, and the funding sources, especially JGI BioFICUS program. With that, I thank you for your attention.